Welcome to my presentation on the differential gene expression analysis of Parkinson disease versus healthy controls. For this mini project, I have analyzed the human dataset GAC 165082 comparing Parkinson's patients to healthy controls using the DSEC2 pipeline on RNA sequence data. I'm Samal Kumar, roll number 123BT 10034. In this project, our main goal was to identify statistically significant differentially expressed genes or DEGs between whole blood RNA samples of Parkinson patients and neurologically healthy controls. To achieve this, we applied the DSEC2 negative binomial model to the raw read counts. We aim to report genes passing a 10% false discovery rate cutoff utilizing APGLM shrunken log to fold changes. Finally, we visualized the data and interpreted these DGs in the context of Parkinson's pathology. The data set GSC 165082 was sourced from the NCBI gene expression omnibus. It contains whole blood transcriptomases from 26 human subjects, 12 diagnosed with Parkinson's, and 14 healthy controls. The data was generated using high throughput pair and RNA sequencing. Out of over 63,000 raw genes features, we retained 25,329 genes for analysis after pre-filtering for a minimum row sum count of 10. After running the complete DSEC2 pipeline with the FDR controlled at 10%, we isolated 11 statistically significant DEGs. Among these, 6 were upregulated in healthy controls and 5 were upregulated in Parkinson patients. While the raw P value showed over 2,800 nominal significant genes, the rigorous FDR correction distilled this down to 11 highly reliable calls. This modest number is actually expected for blood-based biomarker studies of central nervous system disorders. Here is a complete table of those 11 significant DEGs displaying their mapped gene symbols, APGELM shrunken log2 fold changes, and adjusted p-values. You can observe genes like GCNT1 and C2 upregulated in healthy controls, while genes such as APOBEC3A and HMGA1 are significantly upregulated in the PD control. Moving to quality control, this principal component analysis plot was generated using VST transformation data. The red circles represent Parkinson samples and the blue triangles are healthy controls. Principal control 1 captures 25% of the variance, reflecting the primary transcriptomic difference between the two groups. While there is some overlap, which is highly typical for peripheral blood data in neurological conditions, there were no extreme outliers, allowing us to safely retain all 26 samples. This sample-to-sample distance heat map further validates our experimental grouping. Based on Euclidean distances, we can see that the samples cluster broadly by the disease status. Again, some intergroup mixing is present due to subtle biological signal, but the absence of isolated outliers confirm our dataset is clean and ready for differential analysis. To verify that our DSEC2 model fit, we generated a dispersion estimates plot. The black dots represent gene-wise maximum likelihood estimates, which is pulled towards the fitted red trend line to generate the final shrunken estimates, shown in blue. This beautifully fitted graph confirms that DSEC2 correctly models the mean variance relationship inherent in a RNA sequence data and successfully pulls out noisy low count genes to reduce false positives. Next, I have the MA plot comparing the unshrunk log 2 changes on the left with the APGLM shrunken estimates on the right. Significant DEGs are highlighted in blue. Without shrinkage, low mean count genes display extreme, unstable fold changes driven largely by sampling noise. The APGLM shrinkage effectively pulls these noisy estimates towards zero, while leaving high count genes largely unaffected. This dramatically improves the reliability of our downstream visualizations. Here is the volcano plot visualizing our differential expression results. The x-axis shows statistical significance and the y-axis displays the shrunken log to fold change. A strict 10% FDR is marked by the horizontal dashed line. The vast majority of genes fall below this line. reflecting the inherent subtle transchromatic signature of Parkinson's disease in blood. The red points on the right show genes upregulated in healthy controls, while significant genes on the left, such as CXCL1 and HMGA1, are upregulated in Parkinson patients. The heat map displays the Z-score normalized expression values specifically for an 11 significant EEGs. The hierarchical clustering clearly separates the genes into two blocks. Those elevated in Parkinson's, like HSPA8 and HMGA1, and those elevated in healthy controls like GCNT1 and CD86. The consistent expression pattern along most samples within each group demonstrates that our findings are robust biological signals and not just outlier-driven artifacts. So, what do these 11 genes tell us biologically? First, we see the upregulation of XLCL1, CD86, and ApoBEC3A in the PD group. These are established markers of active inflammation and immune activation, supporting the hypothesis that systemic peripheral neuroinflammation accompanies central neurodegeneration. Second, the upregulation of HSPA8 points to a cellular proteostatic stress. HSPA8 encodes a critical chaperone in chaperone-mediated autophagy, a pathway directly impaired by alpha-synuclein. This elevation likely reflects a compensatory cellular response. Finally, the upregulation of HMGA1 suggests global chromatin and epigenetic dysregulation within these blood cells. Furthermore, we observe higher expression of the complement component C2 in healthy controls, implying its suppression in Parkinson's. We also noted two sex-linked genes, XIST and UTY, because XIST is strictly female expressed and UTY is male expressed. Their presence simply reflects a sex imbalance between our sample cohorts. Ideally, we should have control for sex and age in a statistical model, as was done in the original Henderson study. However, because our raw GEO count file lacked a sample level metadata, our simplified model lost some statistical power. Despite this limitation, our top 11 genes align perfectly with the populist literature, confirming that a pipeline works accurately and captured the strongest signals. This slide briefly highlights the core R code used for data loading, pre-filtering, and executing the DSET2 pipeline. The complete reproducible analysis was programmed and executed in a Google Colab notebook using the R kernel. To summarize, we successfully analyzed 26 whole blood RNA sequence samples from the GAC 165082 dataset using DSET2. We identified 11 statistically significant DEGs, 6 upregulated in healthy controls, and 5 in Parkinson patients. These genes converge on critical biological themes of neuroinflammation, proteostatic stress, and chromatic and chromatin dysregulation. Ultimately, these blood-based transcriptomic signatures represent promising candidate peripheral biomarkers for Parkinson's disease that require further validation. Thank you.